Hi, my name is Lex Kalsert, and I'm the Director of Research Development at the National PKU Alliance. As part of our mission to educate, we have produced a series of short videos to help people with PKU understand the rapidly emerging field of gene therapy. Gene therapy is a new approach to treating certain diseases, such as PKU, where the primary cause of the disease is a genetic mutation in the DNA. In the U.S., the development of all new medicines is regulated by the U.S. Food and Drug Administration, or FDA, who sets the path for testing a new drug in humans. The FDA is first focused on safety, followed by demonstration of efficacy, or how well the drug works. When a new drug candidate enters a clinical trial, it is called an investigational drug or an investigational medicine. Investigational medicines are first discovered and evaluated in laboratory studies and animal studies to get an idea of how it might work in humans and whether or not it may be safe enough for human use. If the new investigational medicine passes these tests in animals and the FDA approves the application, it can then be evaluated in humans in clinical trials. A clinical trial is a series of research studies involving groups of people who will be treated with the investigational new medicine under similar conditions, exploring such things as the amount of investigational medicine to be given and how often it may be given. As each question is answered, a new group of volunteers will be chosen for the next step in the clinical trial plan. All clinical trials have guidelines about who can participate. Before joining a clinical trial, a volunteer must qualify for the study. Examples of qualification criteria can include age, gender, the type and stage of disease, previous treatment history, and other medical conditions. So not all patients qualify to participate in a clinical trial. The clinical trial process is generally set out in 3 phases called Phase 1, Phase 2, and Phase 3. The details of each phase are determined by the type of investigational medicine and the type of disease. For some types of drugs, phases can be combined. For example, Phase 1 and Phase 2 studies can be combined in gene therapy trials, so we often hear about clinical trials starting with a Phase 1, Phase 2. Okay. Which is a little different than a classic clinical trial. Phase 1 clinical trials are designed to see if an investigational medicine that is being given to humans for the first time is safe. Although Phase 1 clinical trials are preceded by years of laboratory and animal studies to characterize the safety, there is still uncertainty as to how humans may react. So it is important to proceed with caution. Phase 1 studies typically start with a very low dose of the drug. These initial doses are sometimes so low they are not likely to have any therapeutic effect and are given to a very small number of patients. After the response to each dose is thoroughly evaluated and determined to be safe, a new group of patients will receive the next scheduled higher dose. This process continues dose by dose until either the dose expected to work is reached or a side effect develops that makes it unsafe to test higher doses. Historically, Phase 1 clinical trials used healthy volunteers. However, for gene therapy, the goal is to permanently introduce a new working gene, and there are uncertainties of how an extra copy of a working gene might affect an otherwise healthy person. Therefore, Phase 1 gene therapy trials will enroll patients with the actual disease. This means that scientists can measure both safety and efficacy early in the clinical development process. And these are sometimes called Phase 1, Phase 2 clinical trials. At the conclusion of Phase 1, the group responsible for the investigational new medicine, also called the sponsor, will write a summary report and submit it to the FDA for review. The FDA can accept the report and allow the clinical trial to move to Phase 2, ask for additional phase 1 studies if there are any concerns about safety, or if warranted, stop the clinical studies altogether. For gene therapy, phase 2 clinical trials usually explore several doses, with each dose being evaluated in a new group of patients. Since gene therapy is a single treatment, the goal of phase 2 is to find the dose that both gives the best therapeutic response and the widest margin of safety. One important outcome of phase 2 is to establish the dose that will be used when the new medicine is approved. At the end of phase 2, the drug sponsor will write a summary report and submit it to the FDA for review. The FDA again can accept the report and allow promotion to phase 3. Ask for additional information if there are any concerns about the results, or stop the clinical trial altogether based on safety or lack of efficacy. The primary purpose of a phase 3 clinical trial is to test the best dose determined in phases 1 and 2 in a larger number of patients. Not all medicines work equally well in all patients, and one important objective of phase 3 is to understand the range of therapeutic responses in a larger group of patients. Not all patients experience the same side effects of any given medicine. Therefore, another objective of phase 3 is to determine the frequency and severity of side effects in a larger group of patients. For example, if an adverse response is rare, it may not have been observed in the relatively small number of patients that participated in the phase 1 and phase 2 trials, but could be discovered in the phase 3 trial. After completion of clinical testing, all the data is compiled into a biologics license application, or what we call a BLA, and submitted to the FDA for review. The FDA can approve the investigational drug for marketing, meaning that a physician can give the medicine to patients outside of a clinical study. It can ask for additional data prior to approval, or deny the application altogether. Phase 4 studies occur after the investigational drug has been approved for marketing. The primary objective of phase 4 studies is to study the safety and efficacy characteristics of the newly approved drug in a much larger group of patients. For gene therapy, phase 4 studies will help determine how long the benefits of gene therapy may last. Clinical trials have a very specific inclusion and exclusion criteria, therefore not all patients will qualify to participate in a clinical trial. Clinical trials have very specific schedules starting before you receive the investigational medicine through your very last visit to the clinic. Your doctor will explain all of the steps and the schedule. All clinical trials have some risk of an adverse reaction to the investigational medicine. Patients must consider these risks carefully before entering a clinical trial. Patients have the right and the responsibility to ask all the questions necessary to make them comfortable with the clinical trial process and the associated risks. Once you've had an opportunity to ask questions, you will be required to sign an informed consent form stating that you understand the process, your responsibilities, and the risks associated with the clinical trial. Once accepted into a clinical trial, patients always have the right to drop out of a clinical trial at any time for any reason. I hope you enjoyed learning more about how a clinical trial works in gene therapy. Our next video will discuss safety.